So today we're going to be picking up where we left off, talking about transport, but to really hammer it home, I pulled up an example question about equilibrium potentials and membrane potentials. So we can go over that one more time because I know it can be a little tricky. So you should see a question projected to your screen right now. I'll pull it up as a poll in a minute, but go ahead and think through that. So I can see that opinions on this are actually split. So I'm glad we're going over it right now. So here's how we should be thinking through this question. So we're given a cell and we're told that the cell has a membrane potential right now. So this is the current state of the cell at negative 70 millivolts. So that's where we're hanging out in the cell. We're asked then to track what is happening with sodium and what's happening with potassium. Now, sodium and potassium are each going to have their own different chemical gradients, but they're also going to interact with those charges. So we're going to have a different potassium equilibrium potential and a different sodium equilibrium potential because we have to sum together the electrical driving force and the chemical driving force for each. And it just so happens that the potassium equilibrium potential is negative. Specifically, it's negative 94 millivolts. And our sodium equilibrium potential is positive. So we have a positive 60 equilibrium potential for sodium. So we can see right off the bat that our sodium and our potassium are probably going to interact pretty differently with regards to this cell. So when we're looking at the electrochemical gradient here, so a way we can think about the equilibrium potential is that that ion is trying to pull on that membrane potential until it reaches its preferred value. So potassium wants that cell to go to negative 94. Sodium wants that membrane potential to shoot up all the way to positive 60. So we can use these facts to figure out which way the electrochemical gradient is going to go. So if our cell is at negative 70, I'm going to look first at potassium first because that's the first equilibrium potential we get. So if we're at negative 70, to get to negative 94, with potassium, which is a cation, positively charged ion, it's going to happen with that potassium. We want to get a cell from negative 70 to negative 94. So to become more negative, to reach that potassium equilibrium potential, we'd actually have to have potassium move out of the cell. So if potassium were to move out of the cell, pulling that positive charge out of the cell, that would make the cell more negative. So for those of you who answered B, that's not correct. We do have a pull on potassium. Potassium does want to move. It's not happy with the state of the cell, but it's actually going to be trying to move out of the cell to make it more negative. So that was our example from Monday, actually. Now, once we look at sodium, so our sodium equilibrium potential is positive 60. So positive 60, we're trying to get to positive 60 from negative 70. And sodium is also a cation, so it's also positively charged. In order to get to positive 60, we're going to have to dump a lot more positive charges into that cell, right? So for sodium to win out and get to its equilibrium potential, we'd have to have a lot of sodium move into the cell. So for this question, A is correct. That's the correct direction for our electrochemical gradient, because to make that cell positive, we'd have to move sodium in. So our electrochemical gradients in this situation for sodium and potassium are in opposite directions. The electrochemical gradient for sodium is to move it into the cell. The electrochemical gradient for potassium is to move it out of the cell. And we can see this by looking at their two equilibrium potentials. Okay, Does that kind of make sense to people? Are there questions about this question at this point? Okay. Well, we can practice a couple more of these in the future. I'll try to find some for us to go through on Friday, but you can also look in the back of your book. They have a couple there, so that's a good source to try and practice questions like this. Okay. So, moving back to our actual packet, so our membrane transport part 2 packet. We were talking about diffusion, and so far we had gone through 2 types of passive transport. We'd gone through simple diffusion, we'd gone through part of facilitated diffusion. So we saw that simple diffusion is small, nonpolar, lipid-soluble molecules going directly through the membrane. And we learned that facilitated diffusion has to happen through some type of a membrane protein. Okay. So we saw that there were both carrier proteins we went through. We also have channels. So when we were looking at a carrier protein, I think our example was glucose. We talked about GLUT4. So we saw that a carrier protein for facilitated diffusion has basically a pocket that fits to our molecule that we're trying to get across the membrane. When that molecule fits into this little pocket, the protein changes shape and dumps it into the cell or out of the cell, as the case may be, depending on the gradient we're looking at. Our second type of facilitated diffusion happens through a channel. And the difference here is that our protein in the membrane is different. So our carriers have a physical shape and they have a pocket where our molecule needs to lock in. When we're talking about a channel, we're talking about basically just an open door in the membrane. Now, we'll see in the future that there are ways to close this door. So these doors can have gates on them, right? Like it is an actual door you can shut. But once it's open, it's open. We don't have to attach that molecule to our protein. It just flows straight through. So like a carrier protein, a channel is a transmembrane protein, which means it has a polar side to touch the ECF, a polar side to touch the ICF, and then a non-polar region in the center, which allows molecules to pass through. And it's going to function like a pore. So when we draw them, you can see they look like pores, basically. We can see this hole, this tunnel through the center. Now, when I say they function like a pore, that may seem like just anything could go through, but these proteins do have their own chemical and physical properties. That pore has a specific size. So these channels actually are substance specific, even though we're just forming an open passageway. So we'll see in the future that there are, for example, sodium channels and that there are potassium channels, which means that when they're open, if a sodium channel is open, sodium can flow through, but potassium wouldn't be able to flow through a sodium channel. So they are substance specific, even though it's like an open door. Okay. We have different types of channels according basically to the substance specificity. So a really important one is something called an aquaporin. And I say important because it's important physiologically. So aquaporins specifically allow water through. And we'll see as we move forward that the movement of water is really important to how your body functions. So we know about osmosis. You've heard that word before. So part of that happens through aquaporins. Aquaporins help make membranes permeable to water. But we also have channels that allow other types of molecules through. So our ion channels are also very important. And we'll see that what they largely do is allow for signaling between cells. So we'll see that our neurons function largely through opening and closing ion channels. We'll see different types of ion channels as well. So one thing is that we'll have ion channels for specific ions, like sodium channels and potassium ions. But we'll also have ion channels that open in specific ways. We'll have something called leak channels that basically allow ions to leak through, so it allows us to move towards our equilibrium potential. And we just have a little leakage of that ion, always moving in that direction along the electrochemical gradient. We'll also see we have gated channels. So gated channels will be triggered to open by something else. And in the case of neurons, we'll see that we have voltage-gated channels. So once a membrane potential reaches a specific voltage, that channel will open up. And some channels are bidirectional. So they allow our ions to go out, but they also allow our ions to go in. Okay. But all of these channels, once they're open, just act like open doors, and they allow passive transport to happen, facilitated diffusion. So the thing you want to have cemented in your brain when it comes to channels is that channels are passive. They're not actively pushing anything in one direction or the other. So anytime you see a channel, the movement of our ion or water through that channel is going to be in the direction of its gradient. So chemical gradient or electrochemical gradient, depending on whether we're talking about an ion or we're talking about something nonpolar. Okay. There are factors that affect the transport through ion channels, just like there were factors that affected our rate of simple diffusion and that affected our rate of transport through our carrier proteins. So one is the transport rate of the channel. So basically how fast molecules can get through that channel might depend on their size, various properties of that protein, but each channel will have its own sort of base rate at which it functions, how fast it can get a molecule across. Then the number of channels in the membrane also makes a big difference. So more channels, faster rate of transport. And that's just like our carrier proteins. So just like our other type of facilitated diffusion, if you have more ways to get through that membrane, you go through faster. So that's actually also like our simple diffusion directly across the membrane, where we saw if we have a greater surface area, that's essentially more ways to get through the membrane. So we had faster transport there as well. So our channels, more doors, go through faster. The other thing that does affect our rate is going to be whether those channels are open or closed. So sometimes we'll maybe have a lot of channels, but they're all shut because the voltage isn't right, we'll see. So then we would have slow or no transport because it doesn't help us if we have a channel, but it's closed. Okay. And we're gonna go through some checkpoint questions as we do review on Friday, so I'm gonna save these for later. But I want you to key in right now just to remember as we're going through this that we are thinking about some uncharged substances like glucose. We're also thinking about charged particles like ions. And so we're going to have to be tracking different things depending on the qualities of the molecule, qualities of the particle that we're tracking. So our ions have charges, glucose does not have a charge. So ions have an electrical gradient, but glucose and uncharged molecules do not. Okay. So now we're going to move on to active transport. So the fundamental quality of active transport is that we're moving against a gradient. So if we're tracking our ions, this could be against the electrochemical driving force, right? We take both pieces of our force into account, the chemical gradient, but really the electrochemical gradient as a whole. When we're looking at uncharged particles, we're going to be looking at the chemical gradient. So active transport is always moving in the opposite direction that that molecule wants to move on its own. This means that it's not spontaneous. This is not going to happen on its own. Our particles always, always, always move down their gradients, move with their force on net, without energy. They automatically spread out. They automatically try to reach equilibrium just through their random natural movement. So if we want to highly concentrate some area with a molecule, or if we want to completely empty some area of that molecule, we're going to need to put in energy. We're going to need to do work. Okay. So active transport is not spontaneous and is going to require cellular energy. We are going to be using membrane proteins to move these molecules. And specifically, we're often going to refer to these membrane proteins as pumps because they use energy. So if I tell you that we're looking at a sodium-potassium pump, you should immediately know pump means we're pushing, right? We're pumping on something. So this tells us that we're doing active transport. The fact that it's a pump means we're putting in energy to make movement happen. Okay. So our movement that we're pumping is always going to be against our gradient. So here in this example, we see at the beginning, we have an equal concentration of solutes on side A and side B. So say inside the cell and outside the cell. So under normal circumstances, we're not going to have any net movement of these molecules. We're not going to have any passive transport happening because they're already at equilibrium. They're already evenly spaced out. This is their preferred state. So if we want to change that, if we want to increase the concentration of side B and decrease the concentration of side A, if we want to have net movement of these molecules, we are going to have to use active transport. So this would happen via active transport. So we're going to look at the characteristics of a pump. And specifically, we're going to be looking at our sodium-potassium pump because our sodium-potassium pump really powers a lot of the physiological phenomenon that we're going to see in this course. So it's a really important type of pump, our sodium-potassium pump. So a pump is a membrane protein, and it's going to have specific binding sites, just like our carrier proteins that we saw in facilitated diffusion. But the difference here is that we're going to be moving those molecules, those ions, against their gradients rather than with their gradients. So it is functioning as a transporter. So like our carrier proteins, it's basically a type of carrier protein. But a key difference when we're talking about a pump is that it also actually acts as an enzyme. Specifically, it acts as an enzyme because it breaks down ATP. So the enzymatic portion of our pump breaks down ATP to a phosphate group and ADP. So it releases the energy in that bond between the two final phosphate groups, releases that energy, and that energy goes into the pump. And this is what's going to allow us to push our molecules against their gradients. So the enzymatic portion, it's not saying it acts as any old enzyme breaking down some, glucose molecule or something. Specifically, the enzymatic reaction we're talking about is breaking down ATP. So it can harness that energy from ATP hydrolysis, from breaking down that ATP molecule, to pump these molecules across. So here we see one side of our sodium-potassium pump. So what we see is sodium binding into the pump, and our sodium-potassium pump takes 3 sodium ions. So here we see that the sodium-potassium pump has 3 slots for sodium on the inside of the cell here. It has 3 spots. So this means it's at a certain point, it's going to demonstrate saturation. It's going to be full of sodium. And at that point, we're going to be able to use this energy to move them across. So we're going to have the ATP broken down in order to help us move the sodium. So our sodium would then move to the other side of the cell, and then we'd have this pump facing the opposite direction. we'll see a little bit later what happens when this pump is open towards the extracellular fluid rather than the intracellular fluid. Before we get into that, I want to key us into the difference between primary active transport and secondary active transport, because we're actually going to see two different ways that we can move a particle against its gradient, so in the opposite direction of the driving force. And what we were just tracing with our sodium-potassium pump when we were looking at that example of a pump was primary active transport. So when we have this enzymatic reaction breaking down ATP, that is primary active transport. So if you see ATP, it's being used to push something against its gradient, you're looking at primary active transport. Whenever we're trying to identify what type of transport, we have steps. What we look for first is the gradient of the molecule we're tracking. If we're going with the gradient, so downhill, we're looking at passive transport. If we're going against the gradient, uphill, we're looking at active. And so then our next step is to figure out whether there's ATP or not. So if we're going against the gradient and there's ATP, we're looking at primary active transport. So our energy is coming from breaking down ATP. So we saw this in the case of our sodium-potassium pump. We've looked at the sodiums fitting into these three slots on the intracellular slide of this pump. We break down ATP, and then those sodiums get pushed to the other side using that energy. And we also see that our sodium-potassium pump is then going to revert back to the inside, but before it does that, while it's facing the outside, while we have these pockets facing the outside, when our sodium-potassium pump is oriented towards the cellular fluid, instead of accepting sodium, it now has two pockets for potassium. That's just a fact that's true about the sodium-potassium pump. So when we're facing the extracellular fluid, we have to wait for two potassium to bind in to our pump, and then our protein flips again. As it flips towards the inside, it lets go of this phosphate, and it pulls our potassium into the cell. So our sodium-potassium pump moves 3 sodium out of the cell and 2 potassium into the cell, and this is a cycle. So it's not like it does one half of this sometimes and one half of this the rest of the time. It goes through every single one of these steps every time it functions. Okay. So sodium-potassium pump is a really good example of primary active transport. If we want to think about how to remember that, and we remember how the sodium-potassium pump works, then we know what primary active transport looks like. Secondary active transport in a physiological process is often going to happen after primary active transport, which is part of why we call it secondary active. But we can also think of it as having to use a second molecule or a second gradient. So in secondary active transport, it's still active. We're still tracking a particle that goes against its gradient. But instead of getting the energy from ATP, we're going to get the energy from some other gradient, from some other molecule moving downhill. So here in our slides, it says energy is released from ion diffusion. So what it's saying to us is that, let's say we've had our sodium-potassium pump going for a while. So we've pushed a bunch of sodium out of the cell. We pushed it out of the cell until we had a high number of sodium particles floating around in the extracellular fluid. We can think of this as having stored up energy, and instead of storing the energy in an ATP molecule, we just have it stored in a gradient. It's kind of similar to our cellular respiration, right? For a while, we don't have energy in the form of ATP. We have energy in the form of a hydrogen ion or proton gradient inside the mitochondria, which we then convert to ATP. So sometimes in cells, we're going to see energy in the form of usually a sodium gradient. So energy is going to get released from ion diffusion. So we'll have our sodium then allowed to move into the cell. So it's going to move down its gradient. And as it does that, it's going to pull another molecule with it. But that other molecule will be moving against its gradient. So that's the part that makes this active transport. So if you're looking for secondary active transport, you're always going to look for two particles moving or two molecules moving, and one of them is going to be moving against its gradient, while the other one is moving with its gradient. You're not going to see ATP anywhere. So this specifically is an example of co-transport. And specifically, this is a sodium-linked glucose pump. So it's a glucose pump, so it's actively transporting glucose. The way it's doing this is through secondary active transport, because glucose is moving up its gradient. So here we're saying that we have only a little bit of glucose outside the cell. We have a lot of glucose inside the cell, but we want all the glucose in the cell. We don't want to leave any out here in our extracellular fluid just hanging around, not being used for metabolism. We want to get it all into the cell. So we're going to need to do active transport in order to move against this gradient. But instead of using ATP to fuel our pump, we're going to take advantage of a sodium gradient that has built up. So in this case, there would be a lot of sodium outside the cell, but not that much inside the cell. So the diffusion of sodium, so basically the passive transport of sodium, is going to create movement here, and that movement is going to pull the glucose through. So the pump is actively transporting glucose by taking advantage of the sodium gradient. Another example of secondary active transport is the sodium-linked proton pump. And we're illustrating it here because I want to make sure that you realize that we're not always going to have secondary active transport moving in the same direction. It would be easy if our primary active were always in opposite and our secondary active were always in the same, but that's not true. So our sodium-linked proton pump is an example of counter-transport. You might have seen this referred to as an anti-transporter, so moving in two molecules moving in opposite directions. So this is counter-transport via secondary active transport. So here we're working with the same sodium gradient as in the previous example. So we'd have a lot of sodium outside the cell, not very much sodium inside the cell. So sodium wants to move in, okay? Sodium wants to move in, and that even links into our first question about equilibrium potentials, right? Equilibrium potential for sodium is positive 60. So we're trying to make the inside of that cell more positive. So sodium wants to move into the cell. When we let it move into the cell through this sodium-linked proton pump, this movement of sodium powers the movement of protons, which are the same thing as hydrogen ions, out of the cell. So this is kind of like a revolving door. As sodium moves in, pushes these protons out. This is still secondary active transport, just like our glucose example, because we're not using ATP to move protons against their gradient. We're using the gradient of sodium. Okay. But we know it's active because if we looked at this proton gradient, what we would see is that there were a lot of protons, so a lot of H pluses here outside the cell, not very many inside, but we want to pack them all outside the cell. We want to get rid of all of these intracellular protons. So we're pushing them out, even though that's uphill energetically. This is a convenient table from your book comparing all these different facts that we've gone through about passive transport and active transport. So we've talked about each of these factors. We've talked about the direction of net flux. which is to say whether these particles are moving with their gradient or against their gradient. So this shows you that in simple diffusion and our two types of passive transport, we're moving down the electrochemical gradient. For active transport, we're moving up the electrochemical gradient. Tells us about what type of protein, if any, we're using for these different types of transport. So in simple diffusion, we're going directly across the membrane, so we don't have a protein at all. For our passive transport, we had either ion channels or carrier proteins for our two types of passive transport that aren't diffusion. For active transport, we have pumps, both for primary and secondary active transport. And so you can go through this table as you practice. I would maybe make yourself a table of this except blank and try and fill it out yourself for practice, because it can look a little intimidating, but you do know all these pieces of information. This is just kind of a centralized source. Now we're going to look through a complicated example of how all these things would tie together, right? So when we're looking at physiological processes in the body, we're not really going to be looking at any one ion moving all on its own once. When we're looking at how things move in and out of cells to have functional effects, we're going to see both types of transport happening either simultaneously or in a specific order. So we call this active-passive coordination because we're going to set up gradients using active transport. So we're going to make the inside of the cell, so the intracellular fluid and the extracellular fluid, be different from each other, have some type of a gradient through active transport using energy. And then later we're going to be able to release those gradients to power secondary active transport and also to allow passive transport of things across the membrane when we're using that for things like signaling. So here in this example, the first thing, and it's not numbered, so there was no way for you to know it was first unless you spent a long time looking at this diagram. But the first thing we would look at really here is this sodium-potassium pump. So this sodium-potassium pump is powering the creation of a gradient across this cell. So it's powering the movement of sodium out of the cell. So it's packing a lot of sodium into the extracellular fluid. And it's also packing potassium ions into the cell. So we have a lot of sodium outside, we have a lot of potassium inside. So our sodium-potassium pump, both of these move against a gradient because as we put in energy, we just increase and increase and increase the sodium concentration outside the cell. So after we have this happen, we're then able to allow passive movement of these ions as well. So here we can see channels in the membrane. So here is a sodium channel, here's a potassium channel. Because we've built up a sodium and a potassium gradient across this membrane, when our sodium channels and potassium channels are open, we're going to see movement of sodium and potassium respectively. So because using this pump, we pushed all the sodium out of our cell, now we have a really low concentration of sodium in the cell. If we open a sodium channel, we're going to see the passive movement of sodium into the cell with its electrochemical gradient. If, by contrast, we open a potassium channel, this pump has been building up a potassium gradient where we have a lot of potassium inside the cell, not very much potassium outside the cell. So if we open a channel for passive transport of potassium, we're going to see potassium move from the inside of the cell to the outside of the cell. So if we saw this whole system working together, we'd see that the transport of sodium and potassium, even this passive transport, is kind of the direct responsibility of the action of this pump. If this pump stopped functioning, sodium and potassium would reach equilibrium across the membrane. So we wouldn't see this passive movement of sodium and potassium either, even though neither the passive transport of sodium nor potassium requires energy when we're looking at the level of the protein or of the membrane. So this is just an example, and we'll see a lot of examples like this as we go through specific processes in this course. So you don't need to memorize this specific figure right now, but when we get to processes like this that make neurons work or that make your GI system work, we will talk about how these different proteins and how these different pumps and channels are working together. Okay. As I said, we do some checkpoint questions on Friday. So again, we're going to save these, but these are some really good things to think about. These are a little chunkier questions. So if you get a chance and you've started studying for your exam already, which I hope you have, these are some good ones to try. We're going to do a tiny bit of membrane transport part 3. So I'll give you a second to move to your final packet for this exam. And anything we don't get to in lecture for the exam will not be on your exam. I'll go through and make sure that I don't put in any questions on material we haven't gone over. So don't panic that you have to learn all of membrane transport part 3 on your own if we don't get to it in class. So remove that worry from your mind. So this final packet is on the movement of water specifically. So this ties into transport because transport fundamentally is just the movement of different molecules, different particles across membranes. But the movement of water is specifically important because your body is mostly made of water. We take advantage of the movement of water to power a lot of our body's functions, basically. So in order to understand how water moves by something called osmosis, we need to understand how solute concentrations affect our water balance across the membranes, and also to understand how our water molecules are going to interact with the membranes and the solutes surrounding them. Okay. So you've probably seen the word tonicity before, if you've learned about osmosis before, which I think you all have. But you may be a little bit less familiar with this word osmolarity. We're going to talk a little bit first about the difference between these two things. Okay. So when we're talking about osmosis, we're talking about the diffusion of water through a membrane, across a membrane. So we don't usually draw on the channels that water is going through, but this would be the responsibility of aquaporins, which are a water channel, allow water to move very easily across a membrane. They're present in almost every cell membrane, basically. So osmosis is diffusion, passive movement of water across a membrane. Osmosis is always passive. It's always happening automatically down a water gradient. It's not going to be affected by membrane potentials because while water is polar, it's not charged in the way an ion is. So we don't have to worry about how many millivolts we have inside a cell when we're tracking the movement of water. We're going to be tracking, when we're thinking about osmosis, is just concentrations. However, the tricky thing about osmosis is that instead of tracking the movement of a solute, we're down its gradient. We're tracking the movement of water down a water gradient. And the water gradient is going to be basically the opposite of a solute gradient. So when you have, so you have a beaker of water, right? So you have a beaker of water, then you dissolve some salt in it. We'll often describe the concentration of that beaker, telling us how much of our salt is in the beaker dissolved in the water. So that's fine. That's still how we're going to describe concentrations in anatomy and physiology. But when we're tracking the movement of water, we don't really want to know, we're not really looking at the salt gradient. We want to know about the water. So we need to use that information about the molecules that are dissolved in the water to figure out how much space there is left for water. So tracking a water gradient is kind of like tracking negative space. So in this example, maybe this will help a little bit. So what we're looking at here on the outside of this cell in figure A, on the outside, we have pure water. So we don't have any solutes out here. Okay. So here we have our water concentration, quite high, right? Okay. We have a lot of solutes inside the cell. So our water flow, and basically those solutes take up room that water could be occupying. So our water has a high concentration here in the pure water outside the cell, but inside the cell, where we have these solutes dissolved, our water has a lower concentration. So we'd see a high solute concentration, but we have a lower water concentration. So we'd have water flowing into the cell. Here in part B, we see kind of the opposite. So we see water flowing from an area of higher water concentration to an area of lower water concentration. Actually, I'm realizing this figure is a little confusing, so we're going to go through what osmoles are first before we come back to figures like this. Okay. So when we're talking about osmolarity, we're talking about the total solute concentration of a solution. So when we're talking about osmolarity, we're not going to be looking at just the sodium concentration or just the chloride concentration. We're going to be looking at everything that's in that water, because what we care about is how much water is left, like how much space there is for water in that fluid. So we think about it as a solute particle displacing water. This means that when we're calculating osmoles, we need to think about kind of the chemical properties of the substance we're talking about. So if we have 0.1 molar glucose, so we have 0.1 moles of glucose in our little beaker of water, then we have 0.1 osmoles in that water, right? So if it's glucose, just glucose molecules, we have the same molarity as we have osmolarity. If we talk about salt, so table salt, NaCl, dissolved in this water, we have 0.1 moles of salt, 0.1 moles of NaCl. That's not going to be 0.1 osmoles in a liter of water. So the reason for that is that salt, sodium chloride, dissolves in water. So sodium pulls away from our chloride ions. And those are actually each kind of going to take up space away from the water on their own, and more space than if we had just one molecule, nice and concentrated, nice and compact. So if we're comparing the molarity of sodium chloride to the osmolarity of sodium chloride, we're going to multiply by 2 because of this dissociation of sodium chloride into sodium ions and chloride ions. So 0.1 moles of sodium chloride would be 0.2 osmoles of sodium chloride in 1 liter of water. Okay. So because they pull apart, they're now 2 particles. We're going to say it's 0.2 osmoles, which is twice the number of moles we started with before they pulled apart.